Respiration is the body's way of making energy. In this video, we're going to take a look at glycolysis. The fundamental purpose of glycolysis and other pathways in the respiration cascade is to make ATP. So what is ATP and why is it so crucial? Well, ATP, or adenosine triphosphate, is the energy currency of the cell and is necessary to provide the drive to get reactions going. Glycolysis and the citric acid cycle, or the Krebs cycle, are crucial because we cannot store ATP, and so we need to make it continuously through these processes. These images here show you ADP and ATP, and ATP is formed by adding a phosphate group to the ADP molecule. ATP has a negative Gibbs free energy, which means when it's used up, it provides the energy for other reactions. It helps to make it more spontaneous. Okay, so now let's take a look at glycolysis. Glycolysis is the sequence of reactions in the cytoplasm that converts glucose into pyruvate with the production of a relatively small amount of ATP. It's an inefficient process, but it's a quick way to produce bursts of energy for the body. It comprises 11 reactions catalyzed by 10 enzymes. The fundamental reaction is glucose plus 2 molecules of ATP form 2 molecules of pyruvate plus 4 molecules of ATP. The key characteristics of glucose that's summarized in the figure here is as follows. It's the first step in carbohydrate metabolism. It occurs in the cytosol, or the cytoplasm. From the reaction above, you can see that there's a net production of 2 ATP. So 2 ADP is used up, 4 ADP is produced, and as a result, a net production of 2 ADP. And no oxygen is needed in glycolysis. An anaerobic process is one where no oxygen is required. The first step in glycolysis, out of the 10 different steps that I mentioned, is to ensure that the glucose being acted on cannot leave the cell. So how does that happen? An enzyme known as hexokinase phosphorylates the glucose and traps it in the cell. So phosphorylation, or phosphorylates, means it adds a phosphate group to the glucose molecule, and that's shown in yellow in this figure over here. The extra charge prevents the glucose molecule from exiting the cell membrane. Remember, the cell membrane is hydrophobic, and so a charged molecule cannot easily leave the cell. An ATP is required to provide the phosphate group in this step. In the previous slide, we had seen that 2 ADPs are used in glycolysis, and this is how the first of those 2 ADPs are used. So the next step in glycolysis is a rearrangement of the glucose chain to make it fructose. The third step in glycolysis uses another important enzyme. This one is called phosphofructokinase 1, or PFK1. A kinase enzyme acts to add a phosphate to a molecule. In this case, the molecule is phosphofructose, which is a fructose molecule with a phosphate group attached. Since a kinase enzyme is used to add a phosphate to a phosphofructose molecule, it's known as phosphofructokinase. 1 ATP is used in this process, and now we have a biphosphate fructose, which is a fructose molecule with 2 phosphate molecules. 2 ATPs have been used up till now. This step is an irreversible step, and now the molecule is committed to glycolysis. There's a lot of regulation that goes on in this step regarding how much of the enzymes to make, whether it's phosphofructokinase or the earlier enzyme hexokinase that we saw. For the sake of simplicity, I won't get into those details, but the general concept is that if there are high concentration of metabolites downstream, or there is enough supply of ATP, then less of this enzyme is made. Remember, phosphofructokinase is used up to encourage a molecule to go into glycolysis. If there's no need for glycolysis to produce ATP, lesser of this enzyme is made. This is basically an application of negative feedback. PFK is allosterically affected by different molecules, including ATP and fructose biphosphate. ATP inhibits PFK. This makes sense, as an enough supply of ATP means PFK doesn't need to drive more molecules into glycolysis. Fructose biphosphate activates PFK1. And this also makes sense, as fructose biphosphate wants to be broken down since it's in a state of high energy and produce ATP. Just a brief review on what allosteric regulation is. Allosteric regulation for an enzyme is the binding of a molecule to an allosteric site that enhances or inhibits its activity. That's what this image here shows you, that when an inhibitor is added to the allosteric site, the enzyme is rendered ineffective, whereas when an activator is added, its activity is enhanced. The next steps in glycolysis, steps 4 and 5, work to ensure the formation of glyceraldehyde 3-phosphate, or G3P. In this case, a 6-carbon molecule is broken into 2 3-carbon molecules. Notice how in the first image another 3-carbon molecule is formed, but it gets rearranged to form G3P once again. Again, you don't need to know the details of this conversion. But note that the important 3-carbon molecule is G3P. So as a checkpoint, up till now, we've seen the following. 2 3-carbon molecules are formed from one 6-carbon molecule. 2 ATPs have been used up to phosphorylate the glucose. And no oxygen has been used so far in this process, meaning that it's anaerobic. In steps 6 and 7, each G3P, and remember that there are 2 G3Ps, get oxidized to form NADH and ATP. This NADH will become useful in the later stages of the metabolic process, but for now let's just focus on the ATP. Since each G3P molecule results in the formation of 1 ATP, a total of 2 ATPs are made from the 2 G3P molecules. In the remaining 3 steps of glycolysis, steps 8, 9, and 10, through a series of rearrangements and enzymes, another ATP molecule is formed, and the final product of glycolysis, a 3-carbon pyruvate molecule, is formed. The enzyme pyruvate kinase is responsible for this final ATP formation, and is the third most important enzyme in glycolysis. Remember again that since there are 2 G3P molecules, and since each G3P results in the formation of this ATP and pyruvate, a total of 2 pyruvate molecules and 2 more ATP are produced. As we saw earlier, 2 ATP were used up in the glycolysis process, but since 4 ATP have been produced when you add up the ATP from steps 8, 9, 10 and the previous step, a net production of 2 ATPs are formed. So here's a summary of the entire glycolysis process. Glucose is first converted to fructose biphosphate using 2 ATP, and then the fructose biphosphate is converted to 2 G3P molecules since a 6-carbon is broken into 2 3-carbon molecules, and then from 1 G3P molecule, 1 NADH, 2 ATP, and a pyruvate is formed. The pyruvate molecule that's formed in glycolysis has 3 possible fates. It can either form ethanol through fermentation, it can form lactate or lactic acid, or if oxygen is available, it can go into aerobic stages of the metabolic process using acetyl coenzyme A. The most common option for pyruvate is to form lactate. So why does it form lactate? Lactate's a common option, and the reason for this is that in making lactate, it forms free NAD+ that can be reused to make ATP through glycolysis again. And that's what this image here shows you, is that once the pyruvate molecule is formed and converted to lactate, NAD+ is regenerated that can then be used again in the glycolysis process to form more NADH. In the next video, we'll take a look at the citric acid cycle, or the Krebs cycle, but here's a short and detailed video on glycolysis that summarizes what I've covered in this video.